Hi everyone, this is part 2 of my virus simulator series, and in this part, I'm gonna try to get to the point where we can create our own unidentified genetic objects, and we can code these by editing these black squares here. Then, we can test them out on our little organism here, like this, to see how infectious it is. Is it really deadly? Maybe it spreads a lot but it isn't deadly? There's only one way to find out, and that's by running the simulation. So let's see it happen, but first, we can't do it yet! Why? Well, we gotta lay down a few more of the basic mechanics for how these cells work before we can get to the actual viruses. So with that being said, I want to address some of the major criticisms that people had for my last video, the most overwhelming one being that I didn't make a KH joke, so whatever, I'm Carrie kisses himself. Is this joke gonna plague me for the rest of my YouTube life? I really hope not, but whatever. Okay, so the first actual criticism people had was that this whole simulation was supposed to be the inside of a human body, which is an animal, but I also claimed that these cells had cell walls, which isn't accurate. If you remember from biology class, animal cells only have cell membranes, whereas plant cells have cell membranes surrounded by cell walls. So thank you for pointing that out, because I don't want to be the spreader of misinformation. But I also want to say that there's nothing in the simulation so far that has actually indicated that we're inside an animal. I mean, nothing in the simulation moves at the cellular level, why is that so hard to say? So we could be looking at the inside of an immobile object, not an animal, this could be a plant. So perhaps you're looking at the inside of Leafy, if you know what I mean. But I also want to clarify the main purpose of the simulation, so that it doesn't get lost. The main purpose of this simulation is not to replicate our reality. I mainly wanted to construct the simplest possible alternate reality, in which a foreign virus could inject its own genetic code into a cell, to hijack it to replicate itself. The simulation says nothing about how actual biology works. Why am I in announcer form now? I didn't have— It's similar to how my evolution simulators from 2015 were awful at describing how muscles actually contract and relax to cause movement, but the simulator was still able to exhibit evolution-like behaviors that we could analyze. Capiche? So you could even claim that this viewport is looking inside an alien world in which animals and plants don't even exist, and cells happen to be strangely perfect squares? I mean, that's already kind of weird. So whether or not it's a cell wall or a cell membrane, I don't think it really matters. We're just trying to see viruses do their thing. Thirdly, quite a few people commented on my video talking about these octagonal things, which I called codons in my last video. Some people suggested I call them genes because each one is able to tell the cell to perform an action, which is more analogous to what genes do in the human body. But again, we're kind of looking at an alien world, and I don't think it matters too much. You can also think of these different colors as this world's version of the adenine, cytosine, guanine, thymine. So instead of A, C, T, G, we've got like A, B, C, D, E, F, G, H, I. There's quite a few more, but there's still the smallest level of genetic material that is able to convey information. So I'm going to keep calling them codons. But there were also people in the comments who were actual graduate biology students who mentioned that telomeres do not behave the same way as these black outlines on each codon. So I really appreciate that type of feedback because I know it came from people who are well educated in their fields. And it is helpful to know that I shouldn't call these things telomeres, because telomeres really only shorten when a cell divides, and these cells don't divide. In my simulation, as I've said before, nothing moves. If a cell dies, it's gone. No one's going to replace it. So a better analogy for what these black things are perhaps environmental damage on active DNA, which just happens from it being out in the open. Fourthly, last video I claimed that Java was a compiled language, but it actually compiles into an intermediate binary form called JVM bytecode, so I was technically wrong, sorry everyone. But that's not the subject of this video, so with all those comments having been addressed, let's actually get to finishing covering all the fundamental mechanics of how these cells live, cause we need to get through this fast. So first off, what's this green outer circle? Last video, I explained that the inner circle were the codons or genetic material of the cell. But the outer green circle, it's more of a track for this green triangle to move along. And what's the green triangle? Well, that's going to be what I'm going to call the hand. And the hand is really what performs all of the cell's actions. As you can see right now, the hand can either point outward away from the inside of the cell or inward towards the center of the cell. So right now it's pointing inward and now it's pointing outward. In addition, the hand can be at any rotation along the green track, which means that it can point to any codon along the genetic material. Now when the hand is pointing outward, as it is now, it can perform outward tasks, which include repairing the wall, digesting food, and removing waste. When the hand is pointing inward, as it is right now, it's pointing towards the genetic material of the cell, so it can perform genetic tasks, such as reading and writing codons. Actually, those are the only two tasks. This is all important because if the hand is pointing in the wrong direction when the cell tries to commit an action, such as the hand pointing inward when the cell wants to digest food, then it won't be able to do it. So with that all being said, there should be one thing that's pretty self-explanatory. When you look at the 3 codons that say move hand somewhere, those codons are just moving the hand, obviously. So if it says move hand outward, as this codon does, it flips the hand outward, and that takes a little bit of energy, but it's worth it to enable it to do these actions. When it says move hand inward right here, boop, the hand moves inward. And then when we say move hand to weakest location, as you can see on this codon that's third from the bottom, it's going to rotate the hand along the green track to point at the weakest codon. And the way we measure the weakest codon is by the thickness of these black outlines, which measures degradation. So it will point at whichever codon has the thickest black outlines and is therefore the closest to becoming corrupted. So those are the 3 move hand codons, which I think is pretty fun to see the interpreter go to one of them. Like watch this one hit the magenta one here, and then this guy goes zoop, and then it goes zoop, and then when it hits this magenta one, it's going to go zoop outward, and then yeah, it's like controlling it like a machine. 2 partners in crime. So that explains the green apparatus, but what about this yellow lightning bolt in the center? It's pretty electrifying, so we should find out what it means. Well, the lightning bolt is really just a visualizer for the cell's energy level, which you can also see in this yellow bar on the right. And as you can expect, a cell's energy level is just that cell's ability to do work, and that comes in the form of performing these cellular operations. Which means that if all the energy has run dry, the interpreter actually runs out of energy to spin around the thing. Okay, that's pretty self-explanatory. But how do cells get their energy? Well, that's pretty self-explanatory too, because cells just need to digest food. When the interpreter hits a digest food codon, and there are 2 of them in the default genome, then the hand will see if there are any red particles within the cell's bounds. The cell really only has control within its bounds. And if there is a red food particle, then the hand will shoot a laser at the food particle like it did just there. As this happens, as you can see it's gonna happen soon here, the cell will gain an amount of energy. The amount of energy a cell gains will be a lot if the cell didn't start out with much energy, and it won't gain that much if the cell already had a lot of energy, for the same reason of diminishing returns I mentioned last time. But at this point I should mention that red food particles can permeate the cell membrane or cell wall, whatever it is. As you can see many times, red particles are just passing through squares as if they're nothing, although it does slow them down a bit. Whereas waste particles, which are brown, cannot permeate the cell membrane, so you can see them bounce against the cell wall. And as I've said before, both interactions will damage the cell wall a little bit. Anyway, to demonstrate how important it is for cells to digest their food to stay alive, I'm gonna do what I did last time, and remove those codons from a specific unlucky cell, and see how that cell fares. This guy's gonna be our guinea pig, and I'm gonna say this digest food codon is gonna be replaced with none, none. And as this happens, you can visually see the codon has these completely black octagons, which don't do anything. So we just sit back and wait. And we can also compare it with its neighboring cells, which don't have that deadly mutation. So while the energy levels are still high, the cell is still able to perform all of its other operations, such as repairing the wall or moving the hand around. But with nothing feeding into the energy level, it's just going to drain lower and lower. And you can see this lightning bolt's already getting really small. So, I mean, you already knew this, but size matters. Ugh. Okay, I really don't want to reinforce bad stereotypes. Voiceover carry here. For those curious, every action costs 1.4 energy except for the write command, which costs an additional 1.4 energy per codon written. With that being said, as soon as this lightning bolt hits 0, which is going to happen very soon, the cell does not immediately die. Just like how when a human dies, the corpse doesn't immediately disappear. The corpse just sits there, almost as if nothing's happening. But in this situation, the wall is what determines if a cell is still able to exist. And now that the cell can't perform operations, it can't repair its wall. And the outside elements, which are all the particles whizzing around, still exist. So food particles and waste particles will still interact with that wall, but with no energy, the cell can't maintain it, and it slowly dwindles away. You can see it's getting dangerously thin, and when it hits 0, that wall just bursts. Okay, I was a little early. It bursts. Which is really sad, but that just goes to show the importance of digesting your food. And it left this visible scar on our organism. But what about removing waste? We can see that there are as many remove waste codons, 2, as digest food codons, 2, but it doesn't feel as important to remove waste as digest food, right? Well, first off, when the interpreter hits a remove waste codon, the hand looks for any waste particles inside of the cell and teleports them outside of the cell. Remember that this is a special operation because waste particles aren't typically able to permeate the cell wall. We can take a look at the stat window on the right side, which tells us how many particles of each type are inside each cell. The number of food particles will go up and down because food can randomly drift inside and outside the cell, but the waste particles, you'll see it always be 0, except for right after a cell digests food, it goes up to 1, and then goes back down to 0 as the remove waste codon takes effect. And because of that, if you look at any healthy cell throughout the entire organism, you'll never see more than one waste particle inside of it. I want to take this time to empirically describe why this is so important. So as before, I'm going to have one cell be our experiment where we remove the remove waste codon and see the detrimental effect it has on that cell's life. So this guy, well, I feel kind of bad for traumatizing him because I just killed his neighbor. But this guy, he hasn't seen anything amiss, so he has no idea what's going to happen. He will be oblivious as I remove his remove waste codons. So for quite a while, okay, first of all, you can see that these codons are completely black, so something is missing. But for quite a while, nothing will seem wrong because this cell is still able to digest food to get its energy, and it's able to repair its wall, so the wall won't thin in, but the effect will happen anyway. Because if you look at the stat panel on the right, you can see the number of waste particles is slowly going up. Now there are 6 waste particles in it, whereas its neighbors have 0 to 1. And the problem with that is that the more waste particles there are inside of you, the more collisions those particles will have on the wall, but the cell can only repair its wall so quickly. It's like a constant rate. So eventually the bouncing rate of this accumulating waste will overwhelm the cell's ability to repair the wall, and the health will just continually go down. Well, it forms more of a sawtooth pattern, but when it hits 0, which it didn't quite there, the cell's gonna die. You can think of this as increasing the pressure of poop inside your body, which is kind of gross, but I mean, I guess I colored it brown. So, oof, it just exploded. And waste, not poop, is expelled everywhere. And that basically shows that it's a good idea to go to the bathroom because explosive, never mind. Okay, I think you should already know that. And with that, we've fully explained all of the outward operations, as in all the things the hand can do while it's pointing away from the inside, we've covered. That means the only 2 operations left to understand on this genome are the read and write operations, which point inward to the genome. Voiceover carry here. As a reminder, each individual codon goes through a gradual, somewhat randomized degradation process, and if this is left unchecked, 100% degradation will turn a codon into a defunct none codon that can't do anything. So, as we've already established before, move hand to weakest location, we'll move the hand to the codon with the most degradation, meaning the thickest black outlines. And let's just look at that one more time. So this guy in the upper right looks like it has the weakest, and yeah, the hand just moved there as soon as the interpreter hit the move hand to weakest location codon. So the read codon, what it does is it looks at what the hand is currently pointing at, it looks at it, and it commits it to memory. So you can see that the cell has a memory stat in the lower left, and every cell has its own memory. So this guy is remembering the number 11. This guy is memorizing 6700. Well, now he's memorizing 22 because a new piece of information was written to his memory. And those numbers correspond to the type of codon the hand is looking at. If you want to get really specific here, the meaning of remembering 33 is the codon repair wall. And if you go into this editor, repair is the third option of a starting piece of the codon, and wall is the third possible ending of a codon. So repair wall can be encoded as 33. And that's pretty much how all memory works. Secondly, whenever the interpreter hits the write codon, it's going to take whatever is in its memory and spew it out of the hand, wherever it may be pointing, and it's going to overwrite the cell's genome wherever that hand is pointing. Because the new spewed out genetic material is new, it has no degradation. So what happens is the hand doesn't move between these two read and write codons, so the cell will read from a codon location and then write exactly to it the same location and the same information. So the degradation of that fresh material is 0, so we essentially replace the genome with a fresher version of itself. And this way, if the cell can continuously find the weakest location, pinpoint it, read it, write it afresh, the cell can make sure that no codon ever reaches 100% degradation before it's too late. And that's why, if we zoom out, every single healthy cell has a full genome still intact. So, as I've already done 2 times before, let's do an experiment. Let's choose one of the guys on these islands and say, you can no longer write. Your write codon is gone. And so the cell will continue to do all of its other operations. It can even still read genetic information into memory, but it just can't do anything with that information. And out of all the possible deformities, this one takes the longest effect to wreak havoc on its cell. Because while these codons are still not fully corrupted, it's functionally identical to the other cells. So we really have to wait. Without an ability to repair the degradation on the codons, their health just gets worse and worse. And you can see this black outline just encroaching further and further inward, which is not healthy. So I might fast forward, or I might not. Maybe I'll take this time to explain another small tidbit of information that I glossed over. RGL. I didn't explain what that means. That stands for relative genetic location. And it means if it says 0, that means that we care about the exact codon that the hand is pointing at. If it says -1, then we're talking about the codon 1 step counterclockwise. And if we're talking about like 2, that's 2 codons clockwise of where we currently are. So using these numbers, we can actually describe a range of codons from say -3 to 4, and this will describe multiple codons at once. I'll toy with that later, but for now, you can see that both of them say RGL 0 to 0 and RGL 0 to 0. And what that means is that we're only looking at the codon directly in front of the hand, so we're reading one codon and we're writing one codon. Okay, so you can see a lot of these codons are nearing 100% degradation, so what's going to happen when we reach there? Well, the none codon is 100% degradation, so we're almost there, but the repair wall codon, which happened to degrade the fastest by random chance, is going to turn into a none codon in just a few more seconds, I think. So it was able to repair the wall, but next time it comes around, it might not be able to. 3, 2, 1, almost there. Oh, it did it again. Okay, now it's a none codon. It's fully degraded, which means the next time the interpreter goes around, it won't be able to know what the codon was trying to say, and it'll just do nothing. So there, no wall restoration happened there. And then it's just kind of a domino effect. More codons will get fully degraded, and the cell's ability to perform operations will be worsened and worsened. So now it can't repair its wall at all. And it's pretty clear to see how this will have an effect. But you can see it survived for something like 200 or 300 ticks before anything was amiss, but now things are very amiss. And that is a problem. Okay, I'm going to zoom out a little bit to compare it with its brethren. And yeah, wall health is really bad. Energy's still fine because I guess the digest food... Oh, okay, there it goes. It died. Yeah, so there you go. That's the last of the 3 experiments I need to show you on how crucial these things are. But yeah, now you know how this full genome of 11 codons works. And I think theoretically, with a healthy genome, these cells can live forever. So the idea is, these cells have a genome of 11 codons that allows them to have a perfectly balanced, long-lasting life that you can now fully understand. The rest of this video, and all future videos about this simulation, will be tinkering around with this calm equilibrium and seeing what calamity ensues. I was mentioning relative genetic location earlier, and I just want to do a simple experiment to demonstrate it. So 0 to 0 means that we're only looking at the codon directly in front of us, but 1 to 1 means that we're looking at the single codon, 1 codon clockwise along the path, which means 1 codon in the future. So I'm going to wait for the interpreter to pass, but as soon as it does, I'll change this to RGL 1, 1, like that. And what that means is the hand is going to read the unhealthiest codon and then write it 1 spot over. Do you see that? It wrote this codon's information right here. Which is really bad because now we have the wrong codon in that location and the right codon isn't even healed. So 2 bad things happen. And if this continues to happen, well, I guess this codon will continue to be the weakest, so the hand will just not move from that location ever. But this will also result in cell death. I think this should help paint a picture for how changing those numbers changes how the hand reads and writes information. I guess what I'm saying is that when it comes time to construct a virus, we're going to want to feel comfortable manipulating these ranges to the numbers that are most effective in virology, or fake virology, because we're living in an alien world right now. Okay, so should I wait for the cell to die? I guess I will. Alright, so the first move hand inward codon just died, but that actually has no effect on how the hand does its operations because there's 2 of them in a row. But the digest food codon is now about to die, which means that this cell might actually die from starvation before it dies from wall health decay, I think. Although starvation leads to wall health decay, so all cells die from wall health decay eventually. What is that? Oh my god, okay. I haven't introduced this to you, but this is actually a UGO. I don't know how the cell produced that, but... oh, there's another one. Okay. Yeah, if the hand is pointing in the wrong direction, then when the cell tries to write stuff, it actually produces UGOs, unidentified genetic objects. And, okay, this is really spooky because I didn't intend for this to happen. Oh, there's 3 of them now. Okay. Yeah, this was not part of the lesson plan, and I didn't want to introduce things too quickly, right? I wanted you to help figure out this puzzle yourself, but I guess you're getting an introduction to what will happen in a later video. Okay, now there's 4 of them. Wow, I did not think that... Man, okay. When I created my own virus, I thought it had to be hand... Okay. Okay, first of all, I want to say that viruses in the real world are not manufactured in a lab. That's just a conspiracy that, you know, I don't think we even have the technology to do that yet. Maybe we do, maybe not. But in terms of this simulation, I thought that viruses had to be like so perfectly hand-picked, or not hand-picked, but like they had to be constructed to do the correct thing that I didn't think that a cell would produce its own virus from me just doing a simple experiment. Like I only changed... Okay, wow. Okay, now it's spreading. My god, okay. This was really not what I had anticipated. I even had this whole order of like when the virus, like which viruses I would introduce to you first to show you, oh, this one's not super infectious, but it's deadly, and this one's not super deadly, but it's infectious. But this throws all that out the window. Now I will say, these viruses are not going to be contagious in the sense that the cells that have been infected are going to be fine because the genetic material they inherited is just a none cell, and as long as that doesn't overwrite other genes, it will continue to operate fine, just a little bit slower because it takes longer for the interpreter to go around. Maybe I should keep this experiment going just to see what happens, but I think that no cells will die after this. And wow, okay, we're both learning stuff here because I don't know what's going to happen. But I think I should take this time to just explain what happened. So I told you that a cell's hand has to point outward to perform outward tasks such as digesting food, removing waste, and repairing walls, right? I also said that a cell's hand has to be pointing inward to perform genetic tasks such as reading genes and writing genes, or codons. But I didn't tell you yet that if a hand is pointing outward when it reaches a write codon, the write codon compels it to spew whatever is in its memory out of the hand. So if the hand is pointing outward, then it's going to create an unidentified genetic object outward into the world. So that is what those black dots that were bouncing around were. Those will stay inside of a cell until the cell's wall is destroyed, because the cell doesn't have any mechanism to remove the UGOs. But when the UGOs are now floating freely in the bloodstream, or I guess the plant tubing stream if we're in a plant, because I know plants have those tubes that, what are they called, like xylem or something, that helps move water and nutrients from the roots up into the leaves at the top. Anyway, when the UGOs, which might or might not be functioning viruses, are in the xylem, as soon as they touch a cell wall, I don't really know how to explain this, but that genetic material will instantly glorp itself into the genome at the location the interpreter currently is at. So you can imagine that this neighboring cell, like, okay, so this cell here died, and then the cell wall was removed, so then one of the UGOs just happened to touch the cell wall of this healthy cell and inject itself right here. And the reason why it doesn't just bounce off the cell wall like the waste particles do is because a crucial trait of viruses in our real life that makes them so deadly is that they'll bind with healthy cells and inject their DNA or RNA into the healthy cell. So there needs to be a way for this UGO to touch a cell wall and then have its genetic material inside the cell suddenly. Actually, you know what? I'll just do it now. Okay, I can create UGOs right here. So right now the UGO is just 5 none codons, which does nothing. But if I launch it, let's launch it in this direction. Then, oh no, I don't want to launch 2. I launched 2. Okay, that's bad. So that one just touched that cell and then infused itself. So there's its own genetic material inside the cell. This guy's going to loop over, so let's watch on the other side. Okay, there it is. So pay attention. As soon as it touches the cell wall, its own genetic code of 5 none codons appears within the cell's DNA, because it got injected there. And it was right behind where the interpreter was along the genome. When that happens, I consider the cell to have been tampered with, because it no longer has its God-given genome. Or I should say Carrie-given genome, because I was the one who wrote that. So tampered cells appear a light green, and you can see in the upper right, I have the number of healthy cells, 34, the number of dead cells, 4, and there are 4 dead cells because of I had the 3 experiments to show, you know, what each type of codon does, and the 4th one died through the unexpected viral infection, which is actually not a virus. But I think you should notice that the number of tampered cells is not increasing. It's staying stable at 6. And I think as long as we run this, it will stay that way forever. It's because, as I've already said before, this virus is not infectious. As soon as a cell gets infected, it's able to function like normal and not spread any more genetic material to any neighboring cells. So I can continue to just, well, okay, I'm not going to keep spreading UGOs, but I think I've already made my point that, well, it kind of sucks for these cells themselves because they have less time, as in it takes now 16 ticks to go through one cycle than 11. And what that means is more damage will happen to the cell wall by the time it makes one cycle. So the repair wall will have to do more work to kind of catch up with the battering it had, which is why the cell wall of this guy is thinner than the others. But as long as it doesn't hit 0, which hasn't happened yet, it'll just survive with just a slightly more miserable, thin-walled, thin-skinned, that's the word to use, with a slightly more miserable, thin-skinned life than its brothers. It's like having a benign disease. Future Carrie here. I was gonna fit a couple more viral experiments at the end of this video, but it's already approaching half an hour long, so I'll just put it in part 3. I think I'll make the code open source by putting it on OpenProcessing once I upload part 3, because by then you guys will have seen a fully functioning virus. If you made it this far, feel free to comment any corrections or feedback you've got for this video. Thanks for watching and goodbye!